Okay, first thing, we have to start what is Auchenorrhyncha or Auchenorrhynchos hoppers, because as you see, we have several terms introduced in 19th century by Merrill. In original French spelling it was Auchenorrhyncha, but we have several other. What is interesting, relatively recently, it was a paper, some discussions about the correct date of publication of this name. So according to those two papers by Gregory and Boone, the correct date is 1805, not 1806, as it is still present in vast majority of the papers when the name is used. Of course, beside of the Latinized form Auchenorrhyncha, we have also several other terms for the same group of insects, like Colirostres, Gulerostria, Hypocephalocera, Cicadoptera, all those names based on the genus Cicada, like Cicadarie, Cicadina, etc., etc. So you can see that even with the correct name of the group or groups, we have some problems. Why I'm rather trying to use Auchenorrhynchos hoppers? There are several reasons for that. In fact, we don't have any good character on which we can clearly define the group. In fact, the only character uniting Fulgoromorphans and Cicadomorphans, the modern ones, is placement of the rostrum, of the base of the rostrum in posteroventral area of the head. The other morphological characters, they look similar, but they are not exactly the same. So, in concept, we have the concept is based on the placement of the rostrum. The content, currently we have two infraorders, two subgroups, Fulgoromorpha with 5 superfamilies and 32 families described up to date, and Cicadomorpha with 11 superfamilies and 33 families. So the group is very, very highly differentiated in terms of its morphology, and because of this we have such high amount of taxa, higher families and lower superfamilies, families, and lower level taxa still described and present in the classification of them. Another point is the relationships between Fulgoromorpha and Cicadomorpha. As you see, many proposals have been presented. Sometimes with Fulgoromorpha and Cicadomorpha treated as a sister group, sometimes they are not treated as a sister groups. So it looks that those relationship between Fulgoromorpha, Planthoppers, Cicadomorpha, Leafhoppers, cicadas, Fronthoppers, etc., are still not fully resolved. It is all depending on the set of characters, on the set of morphological characters, on the set of molecular characters, which is taken into consideration to resolve, in attempt to resolve the relationships. The story starts when we add some fossil groups. As you see here, we have more or less well-defined and solved the relationship, including fossil groups in Sternorrhyncha, but in the remaining Hemipteran groups, in the so-called Eucheniptera, those relationships are not solved. We have several groups which are still present in the modern fauna, the lineage like Fulgoromorpha, the lineage like Auchenorrhyncha, the lineage Plepeata, the only lineage of Cicadomorpha still present, Heteroptera, but we have also several extinct lineages which are placed very, very variously. In general, especially those two groups, Ingruomorpha and Stithinopteromorpha, which are placed respectively as a sister group to Auchenorrhyncha and as a sister group to Heteroptera, both those groups are placed in Cicadomorpha. So if we go with this system, Heteroptera inside the Cicadomorpha, we have to have Auchenorrhyncha inside the Cicadomorpha, which is maybe not the best solution, especially with variability of those groups. There are also some other proposals to unite Auchenorrhyncha and Ingruomorpha and Stithinoptera with Heteroptera, those two lineages, to group them together as Prosorrhyncha. And this proposal is also quite well substantiated with some characters we can find from the modern representatives of the groups and from the fossil representatives of those four lineages. So when we add fossil data, when we add more morphological data, in fact, from the fossils, it looks so that our molecular tree of relationship is going to be collapsed. Why we have this? There are several reasons for this thing. If you look on this most recent proposal of chronostratigraphic chart, when you look here, we have the oldest hexapods present in the Devonian. We have the oldest insects present by the upper Devonian. Then we have something which is called Romero Gap. Nothing is preserved as fossils. And in the terminal Mississippian, in the terminal part of the first half of the Carboniferous period, we have the oldest Pterygota. And those winged insects are quite well differentiated. If we look for the oldest hemipterans, we have them in the Pennsylvanian, the middle Pennsylvanian, in the Moscovian, and then we have probably the oldest representatives of Paleorrhyncha, the oldest representatives of Sternorrhyncha, Cicadomorpha, etc., up to those present of the oldest Heteroptera. So fossil record is giving us a little different image of appearance of particular group. Of course, it is something which has to be taken into consideration when we are reconstructing the relationships. So if we go to the fossil record, to the taxa present in the fossil record, for the moment, the oldest representatives of Planthoppers, the oldest representatives of Fulgoromorpha, is Carboniferous fossil named Auchenorrhyncha magnifica from Moscovian of France. More or less a little younger, we have the specimen, the species named Protoprosboles kneleni from Belgium, from Bashkirian. Once it was believed to be the oldest hemipteran. Sometimes some authors suggested that it could be the oldest representative of Cicadomorpha. The same statement was presented also to younger fossil, to Archegriphis crassinervis from Pennsylvania, from Russia, from this part of the world. But it looks that for the moment, any of those proposals are universally accepted. So if we will have quite good, convincing characters of placement of Auchenorrhyncha in Fulgoromorpha, we don't have very good, convincing characters to place some Carboniferous fossils into Cicadomorpha. What was the host plant of those? What was the feeding behavior of those insects, and how they can be related to the other organisms? It looks that at this time, at the very early stages of hemipteral evolution, at the very early stages of Auchenorrhyncha's evolution, Auchenorrhyncha's hoppers evolution, the main source of food were probably seed ferns, the very early conifers, the very early coniferophytes, maybe also some other plants. But probably those insects were primarily feeding not on the tissues as the recent representatives do, but probably on some plant reproductive organs like ovules or seeds. It looks that somewhere at this time, at the very early stages of differentiation, we have this transition into eating. So phloem is the first source of food for all Auchenorrhyncha's hoppers. Probably we have also some first endosymbiotic relationships of those insects. You can find some more data about those endosymbiotic associations, especially about molecular dating of splitting of Sulcia. I will say something more about this a little later, but I'm not very convinced that the first endosymbiont was really Sulcia or Sulcia-like bacteria. In generally speaking, in terms of some of evolutionary mechanisms, we have two major mechanisms described as the mechanism of red queen model and Kurt-Jester model. Red queen model is more related to relationship between various organisms. It is now very well established hypothesis with several models, with several observations confirming those models. The same we have for the Kurt-Jester, but Kurt-Jester, or I prefer to name it also as a Joker hypothesis, it is a mechanism of some catastrophic events. So if you look on the general, those mechanisms of evolutionary process, you can see that we have those two models working in most cases together, depending also on the scale. So if we go to some details on the red queen hypothesis, it was originally a model proposed to describe and to describe the competition between the species, and this competition was believed to be the main driving factor in evolution and in speciation of various groups. Now it is a little more complex hypothesis with several other mechanisms proposed to find those relationships, but it is one of those models which are now very well established in evolutionary theory. The other hypothesis, the Kurt-Jester or the Joker, suggests that if we have catastrophic events, random geological events, climatic events, those events could create the new niches, and those catastrophic events could be the driving force behind the evolution and diversity of the species. It is also a hypothesis which is more established based on the observation of the fossil record, but as you know, fossil record is not the perfect, so we can have some bias in fossil record of various groups. Beside of those two players, we have some other partners in those interrelationship. We have symbiotic bacteria, we have symbiotic organisms. And as you see on those schemes, Sulcia was proposed to be the main endosymbiotic, primary symbiote, the main endosymbiotic bacteria present in all Auchenorrhynchos hoppers, present in both Fulgoromorpha and Cicadomorpha. But we have to have the reservation. We know only the terminal taxa, we know only the model representatives of those groups, and it is now more and more proved that we have several periods, several acts of transfer, horizontal transfer of various endosymbiotic bacteria. So even if Sulcia is highly specialized, especially this kind of Sulcia which is present as a primary strongly reduced genome, it could be also the result of horizontal transfer, not the result of just coevolutionary traits between those groups. Unfortunately, for the fossils, we don't know what kind of endosymbiotic organisms they can have inside. They can have as partners in those coevolutionary traits. But when we analyze the other groups, it looks that we have a high variety of various bacteria, and not only bacteria, also fungi, also yeast-like fungi present as those endosymbiotic partners. So we have a little incongruence between fossil record, molecular record, and endosymbiotic organisms if we try to put everything into a single relationship tree. During the late Carboniferous and early Permian, we have the period of diversification of various plants. Of course, mainly gymnosperm plants, but representing also those groups which are not present in the modern flora, the groups which gone extinct, the groups which are present in the Carboniferous and Permian, or only in those Permians. So those plants were very suitable source of food for many insects, including Auchenorrhynchos hoppers. Because of the architecture, we have also a new ecological niches present in relation with those plants. But as usual, we have some good things with new plants, with new sources, but we have also some limitations, because plants are also trying to defend against different insects. They were producing several mechanisms, defense mechanisms against the insects. We have not only mechanical defensive mechanisms, but also some physiological ones. So all those changes in physiology of the plants were very challenging for the insects, especially for the phytophagous insects, especially for those insects which are relying on the phloem. Also, the presence and the number of the plants was going to be more and more limited, especially in the late Carboniferous and early Permian. It is a period in which we have very high level of oxygen in the atmosphere, but because of this we have wildfires, we have very drying conditions during those geological periods. In the southern part of the world, in the Gondwana, we have even the glaciations. So there are a lot of climatic paleogeographic reasons limiting the availability of the host plants. In fact, you can imagine that all those interiors of the continents, they are just desert. No plants, no water, no soil. So they are really limited, the areas which are suitable for life were limited to more or less coastal areas or near the shores of the rivers. From the Permian, we have some representatives of Fulgoromorpha already diversified, especially representatives of the extinct two superfamilies. Superfamily Coleostecoidea with a single family, Coleostetidae, and Suriococcixioidea with single family, Suriococcicidae. However, for the moment, it is all those taxa placed in Suriococcicidae are placed in the single family, but in near future, this group will be subdivided because there are some venation patterns clearly separating at least some genera which are placed now in Suriococcicidae. When Coleostetidae are limited only to the Permian period, the middle to late Permian, Suriococcicidae survived those periods by the end of those catastrophic events by the end of the Permian, and we have also some Suriococcicidae present in the Triassic. In the Permian times, we have enormous diversification of all those Cicadomorpha lineages, with a lot of groups which are no longer present, with very modified venation patterns, with very modified morphological patterns. And when you look on the right side, you have those Inguomorpha, Stetinopteromorpha, representatives of those groups. Inguomorpha are probably the sister group to Auchenorrhyncha. This is probably the group which is the closest relative of Auchenorrhyncha. Stetinopteromorpha is, it seems it is a paraphyletic assemblage, but somewhere among those groups we have to find the ancestors of the recent Heteroptera. So again, a problem for classification. If we have the groups like Auchenorrhyncha and Heteroptera nested within Cicadomorpha, how to manage with classification, simple classification scheme for those groups. We had a lot of highly modified groups. When you look on the wing of Ignotalidae or Perebolidae, they look like not typical Cicadomorpha with polymerous venation, with a lot of regular or irregular transverse veinlets. But it is a period of this diversification of Cicadomorpha. There are still a lot of new things to be described, to be checked with those groups. Fortunately, maybe we will have some new outcrops with more complete specimens, not just wings. Anyway, for the moment, all those morphological characters are mostly, and classification is mostly based on the venation characters of those groups. Quite recently, we have also finding of Prospolidae, for example, in South Africa, in Karoo. So you can see how diverse those groups were in the late Permian, and sometimes the preservation is excellent, including also the coloration and even the tiny details of the venation which could be observed on the specimen. So, if we go to those Permian diversification period, you can see a lot of lineages like Paleontinoidea, Perebolioidea, Prosboloidea, which gone extinct, and only one lineage is present in the recent fauna. So it looks that we have several models, of which only one model appeared as a successive one. Going back to those evolutionary mechanisms, if we analyze the fossil record of Hemiptera as a whole, including Auchenorrhynchos hoppers, we can find that of those three proposed models of dynamics of the red queen, fluctuating red queen, escalating red queen, and chasing red queen, most of those Permian diversification of hoppers is resulting from escalating red queen and chasing red queen. We have some accumulations of very special characters in those insects. We have a lot of coevolutionary race between the parasites, predators, exploiters, prey. So this is why we have such variety characters in those Permian hoppers. End of the Permian was terminated with the greatest extinction in the history of the Earth. It was the most severe extinction event, during which up to 96% of all marine species and 70% of terrestrial vertebrate species gone extinct. And it is the only known mass extinction of insects. So among those extinct insects, we have also representatives of Planthoppers and Leafhoppers. So the period about 252 million years ago was very, very catastrophic. It was a natural catastrophic event because of large volcanic activity in the Siberia, so we have a lot of consequences of those large volcanic eruptions and lava gases, climatic changes related to those massive eruptions of the volcanoes. So if we look on the results of this volcanic activity, you see the scheme, but when you look at the scheme and you look outside, you have the same results. You have the same changes in climatic zone, in climatic zonation, in climatic conditions as we had in the Permian. Of course, not in such scale, but the mechanism is the same. So the hoppers, other organisms, all insects, were subject of numerous many kill mechanisms. They were clobbered from almost all sides, and this end Permian disaster really reshaped the whole biotic composition and the whole biotic relationships between the organisms, not only in the marine environments but also in the terrestrial environments. So it is the example that we have this competitive race according to the red queen mechanism, but sometimes if we have such a biotic catastrophic event like Joker or Cook Jester mechanism, it could reshape completely the pattern of evolution. After this mass extinction, we have very poor habitats, mostly covered with Pleuromeia, a kind of herbaceous lycopsid, and this plant was not the best source of the food. Unfortunately, we don't have a very good record from the earliest Triassic. We know that we have the recovery of the vegetation, we have recovery of animal life, but it is not very well recorded in the fossils. Among the fossils, it is not very well recorded because of taphonomic bias of the habitats which are present and which are available for animals, for insects during the earliest stages of the Triassic. So, if we look on the fossil record, the Triassic, it is the period in which we have origin of modern Auchenorrhynchas. We have a lot of groups, especially within Cicadomorpha, which gone extinct, and we have one successive group which started to originate, started to diversify during the Triassic. Of course, Triassic was quite a long period. We have also several global changes. We have also some reshaping in the food landscape, reshaping of the vegetation, reshaping in the host plants available as a source of the food. Of course, climatic changes with some cooling events and with some warming events. Anyway, those groups which survive those end Permian catastrophic events, they can start to recover their diversity. They can start to adapt to new habitats, new niches. So they were facing a lot of new challenges. From this period, from middle to late Triassic, for example, we have highly diverse representatives of Goromorpha, of Suliococcixioidea. You can see those variability not only in the venation pattern, but also in the size, also in the shape of the wings. In most cases, we have quite well preserved specimen relatively recently. Even 10 years ago, we had just a few localities like Madigan, like some exceptional localities in the Triassic. Now we have several new Triassic localities which are yielding a lot of fossil insects, so probably the classification and the knowledge of the diversity and disparity of those groups will be much better in near future. In this Triassic period, we have diversification not only of those Cicadomorpha which are present in the current fauna, the Clepeata, but also we have diversification of various non-Clepeata Cicadomorpha. What is interesting, we have the first trace of sound producing by the insects from the Triassic, from the representatives of Dysmorphoptilidae. You can see this. You can see here. It is a very special sculpture present on the ventral side of the wing of the tegmen, and we have a kind of the sculpture on the thorax. So those Dysmorphoptilids were probably the first insects which were able to produce the sound. Some forms of the Triassic non-Clepeata were very bizarre, for example, like those Dysmorphoptilids with very oddly shaped tegmina. It looks that those were mimicking some kind of leaves, some kind of plants on which they were probably hiding on. From the Triassic, we have also some forms superficially resembling, for example, cicadas like Dunstanidae and the first Paleontinidae, for example, like that one from Korea. Non-Clepeata, especially family Maguviopseidae, was extremely highly diversified during the Triassic. Here you have some examples just from a single family, just from Maguviopseidae, with recently described numerous subfamilies, tribes, and a lot of new genera, with various patterning of venation, with various patterns on the sculpture of the tegmina, with various level of sclerotization of the tegmina. Sometimes the specimens are preserved more complete with some body structures. Unfortunately, it is still not very well described. Those body structures are not very well described. Here you have some more forms of those Maguviopseids with oddly shaped wings, and this tendency observed in some Dysmorphoptilids with additional lobes or additional structures present on the wing. We can also observe those in Maguviopseidae. In the Triassic, also some groups which are not very well placed in the current models of classification were also present, and those forms were also diversified. For example, here we have Paranitidae, representatives of Stectinopteromorpha, and this group is believed to be the ancestral to the modern Heteroptera. We have some groups in which it's very difficult to place them in any of those groups currently recognized. So we have some Incertasedis groups, Incertasedis taxa, like for example this Hesederma. Ipsicidae, it is another group with very highly simplified and modified venation. So we can recognize those as representatives of Stectinopteromorpha, but the relationship of this family within Stectinopteromorpha and relationship of this family with Cicadomorpha in broad sense is still not very well understood. Here you have another example of some special characters present in some Maguviopseidae, for example, like those lateral carination on the pronotum, which exceptional for Cicadomorpha in broad sense. Triassic was the period in which we have the first representatives of Clepeata. Clepeata are the only lineage of Cicadomorpha which is successful and present and highly diverse in the modern fauna. Why they have this name Clepeata? The name Clepeata is derived from the Clepeus. They have enlarged, very strongly enlarged frontoclepeus, swollen with distinct salivary muscle traces, and it is the first group for which we suppose that they shift their source of food from phloem into xylem. Of course, it was very challenging, not only because of the nutritional value of the xylem, but also because of some physical and physics things which are related with the pressure in the xylem tissue. With this group, we have also shifting on the median cells into more dorsal position, and it looks that Clepeata is the first group which started to jump. So Clepeata are the real first hoppers. Why they started to shift the food? If you remember, the phloem has a high content of sugar, but not very high content of all amino acids necessary, or steroids, other nitrogen compounds which are necessary for the insects to survive. Xylem is not the best source, but as usual in those cases, with help of some symbiotic bacteria, this difficulty, this challenge could be taken, and it looks that search of nitrogen was one of those triggers for diversification of Clepeata. Especially that in this time when Clepeata appeared in the fossil record, we have some new types of plants appearing in the fossil record. The first more modern gymnosperm plants, but as those plants, as the other plants present from the earlier periods, they adopted a lot of new mechanisms of protection against the pests, against the insects. So from the Triassic times, we have the first true resins present in higher amounts, And it looks that at this time the physiology of numerous plant groups, of numerous, especially gymnosperms, they started to produce a lot of terpenoids, a lot of different chemical compounds present in the resins. On the other hand, we know that also in this Triassic period we have the new types of mycorrhiza between the plants and the mycorrhizal fungi. This also reshaped and changed the physiology of the plants. In this period, in the Triassic, we have also a rapid development of various Auchenorrhyncha lineages, which are competitive for the source of food which was flowing. So maybe shifting to xylem was also one of the methods to avoid the strong competition from Auchenorrhynchas. And at the same time, we have the rapid diversification of the earliest forms of Heteroptera, mostly predatory forms, but they also were looking for some better suitable nitrogen source. So it looks that with those Clypeata we have probably the first endosymbiotic association with Sulcia, so not in the Carboniferous but in the Triassic. It is still not confirmed if it is really a phylogenetic burden or just a result of horizontal transfer of bacteria, but for the moment, the more parsimonious, the most parsimonious solution is that those bacteria were rather a result of the horizontal transfer, not the ancient relationship with Planthoppers and Clypeata, so Cicadomorpha Clypeata, this lineage. Clypeata in the Triassic also started to be very, very diverse. We have representatives of Mesoiaplonidae. Sometimes they look not like typical Cicadomorpha. At first glance, they are resembling even some Frugoromorphans. We have some groups originally placed in completely different insect order, like Mesoiablonia, the whole family Mesoiablonidae. It was transferred to the Hemiptera after the reanalysis of the fossil material. And since the Triassic, we have representatives of Archaeacidae, so the oldest representatives of the current Cicadelloidea or Membrachoidea in broad sense, like for example this fossil Mesoiassus ipsficiensis from the Triassic of Australia. So Clypeata, that's this only lineage which is present still in the current fauna, highly diversified. I don't remember exact figure how many species are already described. I just remember some rough estimation that we know about 20,000 species and more than 100,000 species are still not described, especially those largest groups like Deltocephaline or Typhlosimine. So it looks that those insects are really very, very successful in the modern world. But when you look on this simplified tree of the relationship of various Clypeata, you can see that those groups started to diversify in the Triassic, and they are representing various evolutionary lineages with some terminal taxa highly well established based on morphology, but also supported with some molecular relationship proposals. So, Chelicellidae is the group which is believed to be the most basal, the most ancestral groups to all other lineages of Clepeata present in the recent fauna. So insects with venation, presenting in those schemes, they are believed to be ancestral to singing cicadas, to froghopers, to leafhoppers, to treehoppers as well. So you can see the level of variability. If you compare the level of variability of the modern representatives with this relatively simple venation pattern, it's unbelievable how those insects are so diverse and how they diversify from such relatively simple venation patterns. Why the Triassic is also very special? It is very special period because of another catastrophic event, but it was very long-term event. The event was called Carnian pluvial episode. Would you like to have a constant rain for 100,000 years? Maybe not, but such climatic conditions happened during the Carnian. This is why it is called the pluvial episode. So we have a total rebuilding of the global biota because of high level of humidity, high level of rain during this period. And we can observe the results of this pluvial episode in fossil record, but also we can observe the result of this pluvial period in the modern fauna. Everyone loves dinosaurs, and from Carnian, from pluvial episode, it is the period we have the first true dinosaurs. So it was one of the triggers to diversification of the dinosaurs. But not only dinosaurs, it was a trigger for diversification of modern gymnosperm lineages. It was a trigger for diversification of numerous insect lineages, including Pulgoromorpha, including Cicadomorpha, Clepeata especially. So it was a relatively short period, but it was changing a lot in the global biota. The plants have to develop another mechanism against the fungi. They started to produce a new secondary metabolites, resins as a protective agents, but this change in physiology of the plants was also challenging for the plant-eating insects, including the hoppers, including those representatives of Pulgoromorphans and Clepeata and the other Cicadomorpha groups as well. So it could be also one of the reasons that Clepeata started to feed on xylem, not on phloem. But from the same times, from the same period, we have also diversification of some other groups of Cicadomorpha, for example, paleontinids. They started to develop, they started to diversify during this Carnian pluvial episode. We have also the traces of radiation of Cetinopteromorpha, so this group which is paraphyletic but ancestral to Heteroptera, still phytophagous, still probably phloem feeding, but starting to be more scavenging and looking for new resources of nitrogen. Regarding the other hemipterans, we have probably also a lot of changes, especially in aphids, especially in scale insects. This is the time during which those groups started to diversify, started to present the characters which we can still find in the modern representatives. And it seems that it was the period during which this sulcia, or being more correct in nomenclature of bacteria, Candidatus Carelsulcia Mülleri, this is now current classification of this organism. Probably it started to be more and more related to some groups of hoppers, and because it was universally present with various plants, I think these infections and further recruitment, replacement of co-residents, other bacteria present in the Triassic forms of both Fulgoromorphan and Cicadomorphan lineages is very, very probable. And it looks that this bacteria is also very, let's say, friendly to the other residents, to the co-residing bacteria, which are giving some other resources, which are giving some other chemical products suitable for the host. So I think it's just my opinion that we have to look for the close relationship of Sulcia and hoppers more or less in the Triassic, not back to the Carboniferous as some authors suggested. To explain some of those relationships between endosymbionts and insects, endosymbionts and their hosts, there were some other hypotheses presented. For example, red king hypothesis presented about 20 years ago. So it is the hypothesis which suggests that sometimes the rapid evolutionary change could be a little slowed down because of the coevolution with some endosymbiotic bacteria. This could be this privilege of being a little outside of the control of the red queen models, red queen mechanisms. Unfortunately, for the moment, it is not very well confirmed or rejected in respect to the hoppers. There are no such data available for the Planthoppers or for the Leafhoppers or other Clipper'S. Quite recently, another hypothesis was presented, and this hypothesis is called Black Queen hypothesis. So it is again a hypothesis explaining the mechanism of coevolutionary actions and with microbiome, with bacterial and the host. Again, it looks that at least for the Sulcia, this hypothesis could explain the extreme short genome of the Candidatus sulcia, but again, it is not confirmed with very detailed examination of those mechanisms in the real relationships between hoppers and sulcia or Candidatus sulcia bacteria. Anyway, it looks that this mechanism could be one of the quite good explanation.
My name is Joshua Jones. I'm with the USDA in the U.S. And I'm also a research entomologist on numerous groups, including Psychedelity. And I've really enjoyed the presentation. Thank you so much. I was trying to keep up. I enjoyed these kinds of presentations because you provide a tremendous amount of context, ecological context, to the evolutionary questions of phylogeny, the fossil content and so forth is very interesting. If I understood correctly, and maybe I did not, it sounds like all the non-clipiate forms went extinct in the Triassic, or many of them were culled in the Triassic. Is that correct?
Most of those Cicadomorpha or extinct Cicadomorpha gone to extinct in the Permian. But some groups survived in the Triassic and go on to be extinct in the Jurassic. Some of them even survived to the Cretaceous. So it is all depending on the level on which you are considering those extinction events. We have, for example, families present since the Triassic to the middle of the Cretaceous. We have families present in the Triassic and Jurassic, and they gone extinct by the middle of the Jurassic. Even in Cicadomorpha, similar images with Fulgoromorpha. But it could be also a bias of fossil records. So
sure.
I will not be surprised if we will find something which was not present or not recorded in the Jurassic, finally in the Cretaceous, as it was the case of Fulgoromorpha.
Okay, great. And that's the part of your title on bottlenecks. So this is all very interesting. Thank you so much.
You're welcome.
So are there any other questions? So we'd like to continue. Dr. Szwedo?
Yeah.
Okay.
Okay, you I hope you can see the slide. So I can go a little further with those things.
We cannot see the slides yet.
So I was talking about part of the Fulgoromorpha. For the moment, we have two families present in the Jurassic, Fulgoromorpha and Chiantidae. Yeah?
We cannot see your slides yet.
You don't see the slide?
No, sir.
Yes, sir.
Wait a moment. You can see now?
Yes, thank you.
Okay. So the family Fulgoromorpha is one of the largest nightmare with about 150 species described, of which 1/3 is probably not even a planthopper. But we will see. This is family. This is the group in need of the very urgent revision. So we will see. Maybe the number of the taxa will be reduced to some more reasonable numbers. If you look on the venation patterns of those, you can see that again we have relatively simple. But when you look on the original description or on the recently published catalog of Mesozoic Planthoppers, you will see how nightmarish those venation patterns could be. Unfortunately, we don't have very much whole body fossils. For example, you have here a residual from Upper Jurassic of Karatau. Take a look on this extremely long rostrum, which is also present in some Cretaceous fossils. So it looks, for some reasons, the rostrum should be very long in those times in the Jurassic and later in the Cretaceous. The Jurassic is also another period of another revolution. It is called Mesozoic lacustrine revolution. So we have diversification of aquatic microphytes and diversification of aquatic insects. So we have much more complex trophic webs, food webs, as you can see on this scheme. It is based on one of the localities. It is not maybe directly changing a lot for the Planthoppers and for the Clipeata, for the Cicadomorpha, but in fact we have much better fossil record because of those lacustrine environments of the groups like Helicelidae with quite peculiar venation patterns. We have quite rich record of Procercopidae, the group which is believed to be ancestral to the recent froghoppers, mostly in the northern hemisphere, and probably also because of those new habitats near the ponds, near the lakes. We have also the first fossil record of copulation in froghoppers population of Procercopidae, which is depicted here. From the Jurassic, we have also some extinct groups, extinct Clepeata groups like family Sinoalidae. The first fossils were found in the Middle Jurassic of the China, but relatively recently, more fossils were found also in the Middle Cretaceous. There are no such reasons, but Mid Cretaceous amber from Myanmar, amber from Kachin. So this group was very, very highly diversified, not only in the Jurassic, but for the moment we have all fossils of Sinoalidae from the Jurassic of China, and in the Cretaceous from Burmese amber. Cicadopros is another group which is under-elaborated. You can remember that in most classifications, Cicadopros are treated as subfamily of hairy cicadas of Tetigactidae, but in my opinion, they have to be resurrected as a valid family. There are some morphological peculiarities of those which are clearly separating them, and it is also highly internally diversified group present since the Triassic, quite relatively abundant and diverse in the Jurassic, present also in the Cretaceous. And since these Jurassic times, these Middle Jurassic times, we have the first ancestors of Leafhoppers, family Archaeacidae, in which you can see this character, which is believed to be the character constituting those lineages, fusion of media posterior 3 and 4 and cubitus anterior 1, this anastomosis, which is developed in various degrees in the fossils. Especially you can see the first result of this fusion in Carayasus. So it looks that this group started to be more and more similar to the modern Leafhoppers. Probably in the Jurassic, they started to have spinulation on the legs, and they started to jump. So we have again the first true Leafhoppers somewhere from the Jurassic. The Cretaceous is another challenge for all Auchenorrhynchos, hoppers, and other insects, because we have a transition from the gymnosperm-dominated world into angiosperm-dominated world. If you look here, this is the period in which we have all those odd forms from Kachin amber, terminal Albion, early Cenomania. So you see it is exactly this period in which we have the radiation of the modern lineages of the angiosperms. How old are angiosperms? I will not answer, because some people are even find the traces of angiosperm-like plants even in the Carboniferous, which I believe it is a little exaggerated, but probably this group, this lineage of plants was present in the Jurassic, but in more ephemeral habitats in harsh conditions, and for some reason they started to dominate the all terrestrial habitats somewhere in the middle of the Cretaceous. So this Cretaceous terrestrial revolution, it is a description of very, very intense diversification of plants, especially of angiosperms, but also other organisms, terrestrial, vertebrates, reptiles, birds, mammals, but of course also insects. We have plenty of various groups of hoppers present during this Cretaceous terrestrial evolution. For example, here you have the oldest representatives of subfamily Ledrine. You have some old forms of Fulgoromorpha, but also the extinct group of Cicadomorpha of Helicelloidea, the family Lageronidae, present for the moment only in amber from Kachin. Together with this diversification of angiosperms, we have another very, very important revolutionary event in the Cretaceous. We have rapid diversification of parasitoids. So this period, which started in the early Jurassic, was in full in very, very strong influence to the formation of the faunistic complexes in the Cretaceous. So this mid-Mesozoic parasitoid revolution was another challenge for all surviving lineages of the hoppers, and this could be also the reason why some Cicadomorpha lineages gone extinct. Here it is just a very simplified network of relationship between some groups of parasitoids and hoppers treated as a whole. Regarding Fulgoromorpha, we have not Jurassic, we have also Cretaceous part, especially all those strange forms described and present in amber from Myanmar, like Perforicidae, Dorytocidae, Ubicentidae, also some groups which are present in Fulgoridae, which are classified currently in Fulgoridioidea, like Dorytocidae, Noderbidae, Catlacidae, and we have also Lalacidae, which is now placed in Delphacoidea. From the Cretaceous, we have some very peculiar forms, for example, Neazonidae and Mimarachnidae. Mimarachnidae are very, very diversified among the inclusions in Kachin amber. Neazonidae is a family for the moment known only, and it is very difficult to find the adult which will match those characters present in those names. So if you look on the assortment of various Planthoppers groups present in the Cretaceous, it is enormous. Probably there will be much more units, new taxa to be described from this Cretaceous part, especially from Habil. If we go to cicadas, I already mentioned that we have to have Cicadoprosbolidae again as a valid family. This is the most recent result of analysis, including fossils and including morphological characters present in the large material of cicada and cicada-like fossils from the Jurassic and the Cretaceous, including the nymphal characters. So it looks that maybe in nymphs we can find some key or at least suggestion for the better reconstruction of relationship. I mentioned already Sinoalidae from the Jurassic, but it looks that we have a lot of Sinoalidae still present in the middle of the Cretaceous in Kachin amber. So it's very surprising that we have highly differentiated group present only in two localities, one Jurassic, one Cretaceous, all in Asia. Yeah, that's the proposal for relationship of the modern Clepeata and some other groups. So you can see this image of relationship is going to be more and more complex, especially when we include some fossil groups present in fossil resins like Burmese amber, Baltic amber, Dominican amber, etc., etc. Cenozoic is another period which presented a lot of challenges, a lot of boons also for the insects. Here you have represent some nymphs and a few adults present in some resins. Take a look here. It is the first proof that those nymphs started to jump somewhere in the Paleocene, somewhere in the Eocene. That's the fossil from the Eocene, and you can see those girls which are typical for the nymphs of the recent Planthoppers. Leafhoppers, it could be another 2 or 3 hours to say something more about those groups, but you can see also a lot of forms present in fossilized resins, but also a lot of forms which need to be revised, described from the accretion fossils, from the sedimentary rocks, and of course a lot of new fossils which are waiting for the formal descriptions. Going back to those associations, Cenozoic or Paleocene, Eocene, those are periods in which we have a lot of shifts of relationship between bacteria, between yeast or fungal symbionts replacing the bacteria, as it is case of the Delphasidae. Probably it is one of the adaptations to feeding on monocots, maybe to feeding on grass. So we have a lot of things to be discovered from those groups, especially when you look on this radiation of the grass, and if you make just the scheme, which groups of current leafhoppers are feeding on planthoppers, leafhoppers are feeding on grass, it looks we need some more coevolutionary approach to those events during the Paleocene, Eocene, and later Oligocene, Miocene, when we have the first large areas covered with grass and hemp. So this over 300 million years story is really very, very, very complex. And if we go to the modern diversity is relatively, but the roots of this diversity are very, very deep in the past. Thank you.
Thank you very much, Dr.